Hey everybody, this is Noah Giltner from Microbiology 130, and I did my research project on brain-eating amoeba. So let's get right into it. Primary amoebic meningoencephalitis is the disease that is caused by these brain-eating amoeba, which are called Naegleria fowleri. Primary amoebic meningoencephalitis, as the name would state, is essentially an infection of the brain tissue. The acronym for PAM is PAM. We'll be calling it that for the rest of the presentation. It's a pretty rare disease. Only 145 cases have happened in the United States from 1962 until 2018. Unfortunately, though, of those 145, only 4 people have survived. We become infected with Naegleria fowleri when contaminated water enters the nose and the amoeba migrates to the brain along the olfactory nerve. Infections tend to happen when humans are swimming in freshwater bodies, and these are primarily in southern tiered states. There have also been cases of people getting infected with Naegleria fowleri from using neti pots and nasal irrigation, as well as from swimming in contaminated pools. Primary symptoms are severe frontal headache, fever, nausea, and vomiting. Secondary symptoms are stiff neck, seizures, altered mental status, hallucinations, and coma. As we stated before, mortality rates are really high and people die within 1 to 18 days after symptoms begin. PAM is really hard to diagnose because of the quick onset of symptoms and the quick death. In fact, about 75% of people are diagnosed post-mortem with the disease. So inflammation and lesions, here we have a picture with those white arrows pointing to the frontal lobe of the patient's brain, and you can see it does not look healthy. So let's talk about what amoeba are. And I don't know if you recognize those two ladies, but those are the amoeba sisters who've been helping us out with microbiology all semester. In general, amoeba are large protozoans. They're single-celled organisms from the genus Protozoa, phylum Sarcodina, and they are known for having their pseudopodia, which are their fake feet that they use to move around, as well as to get food and perform other functions. Amoeba in general like to live in moist environments, like soil at the bottom of lake beds, in freshwater bodies, salt water, as well as in animals as parasites and even humans as well. So here's a picture of Naegleria fowleri themselves, the only known type of Naegleria or amoeba that is known to infect human beings. Once they get into your nasal passages, they make your way up to the brain and they start to eat it. So let's look at the cell structure. They are single-celled organisms, and around that cell membrane, the cell membrane itself surrounds the nucleus, which is like the brain of the organism. Cell membrane is filled with cytoplasm, and they also have other other structures inside, such as mitochondria and food vacuoles. You can see here we have 3 different states of the same amoeba. To the left is the cyst, which is when the amoeba itself is being threatened or it feels that it's not going to be able to survive. It creates a thick membrane cell, and it will start to proliferate and clone itself, waiting for the right time for it to come back. The middle state is the trophozoite state, and that is the state when the amoeba will actually infect human beings or other animals. It's kind of when it's at a nice place with food. Typically, Naegleria fowleri eat bacteria inside of fresh water. But if food gets scarce, it moves into the flagellate state all the way to the right, and as you can see, it changes its cell form, waiting for the conditions to return to normal so that it can kind of get back to things like eating bacteria or brains. Here we go, we've got the life cycle here. In the center, you can see a fully grown amoeba. we have the pseudopodia as well as the nucleus. Typically, excuse me, typically, binary fission is the means by which the amoeba will retract its pseudopodia and divide the nucleus by mitosis, essentially creating two clones of itself. Multiple fission does happen when the amoeba is in cyst form, and it makes tons and tons and tons of replicas of itself and its DNA. And when it's feeling like it's ready to go back into the world and conditions get favorable again, the cyst ruptures and lots of amoeba are released out into the world. So here we go. And once again, just a real quick picture. To the right, you see the cyst, which is not infectious. To the left, you see the flagellate, which is not infectious. However, flagellates are found in cerebral spinal fluid of infected people who suffer from PAM. And to the top, you can see when it's in its trophozoic state, which is where it is most infectious for us human beings. All right. So detection and treatment. We mentioned cerebral spinal fluid, and that is one way that they can go ahead and look at it. So direct visualization, if they take a spinal tap, they can actually see the protozoa swimming in your spinal fluid. Antigen detection. Our body does make specific antibodies when the protozoa are present inside of our body. And so with that, coupled with secondary antigen detection, they can use blotting methods to detect the presence of the protozoa. um, PCR is DNA testing. However, we don't have that much information on the Naegleria fowleri, so it's not typically done as much unless it's done under study. And finally, culture, which is a pretty interesting way to do it. They'll take the serum sample, and they'll expose it to a plate that is full of bacteria that the Naegleria fowleri eat. And then they'll expose the plate to incubation temperatures of about 108 degrees Fahrenheit. At this point, most other amoeba will die off, and the heat-loving Naegleria fowleri will survive. Um, as far as treatment goes, early detection is super important. The quicker you can figure out what you've got with this one, the more likely you are to survive. Miltefosine is a drug that's used in conjunction with breast cancer drugs in order to treat the inflammation as well as the infection. Miltefosine is typically used for leishmaniasis, which is where people get bitten by sand ticks and they end up getting these huge, gnarly open sores and ulcers on their skin. So miltefosine is used for parasitic infections. And as you can see, vomiting and diarrhea in up to 60% of patients happens, so it's not a pretty drug. Moving forward, let's look here. From 1962 up until 2018, we can see all of the infections that have happened per year. 1980 wasn't a great year. But from 1962 all the way up until 2013, all but one person had survived. And then in 2013, we have two survivors, and in 2016, there is one survivor. So really quickly, we'll talk about those. The 2013 were two kids. It was a 12-year-old girl and an 8-year-old boy. Then again in 2016, a 16-year-old boy survived the infection. We'll start off with the 12-year-old girl, and she actually got a full recovery. She was diagnosed and treated within 30 hours of infection. She went under the drug protocols, and she also received therapeutic hypothermia. Being that Naegleria fowleri is a thermophilic or heat-loving organism, bringing her body temperatures down below normal, caused her ability to recover, and it damaged or halted the spreading of the protozoa itself. Second case was the 8-year-old boy, and he did end up with permanent brain damage. He was only diagnosed 7 days after infection, which is way too long. He did go under the drug protocols, but he did not get exposed to the therapeutic hypothermia. The 2016 patient also received a full recovery. He was diagnosed within a few hours of getting the protozoa, or the amoeba, excuse me, up his nose. He went under the same protocol as a 12-year-old girl, including the therapeutic hypothermia, and he recovered without any neurological damage. Once again, here's a quick picture of all of the states where this happens, and as you can see, warm states like Florida and Texas, Arizona, Georgia, South Carolina, North Carolina, they tend to have more cases of these um, outbreaks. There was also a 2018 December, so that was just recently. There was a mixed diagnosis. A woman was a breast cancer survivor. Her doctor did see lesions in her nasal cavity, but he suspected that it was a tumor in her brain. When they got up there to do the surgery, her frontal lobe was essentially just mush because she had had the infection so long. And you see that ridiculous picture because it's suspected that she got it because she was doing nasal irrigation or neti pot without boiling her water or using sterilized water. She was just doing it right out of the tap. So think twice before you pour water down your nose. Anyways, I hope you all found that interesting. I hope you all have a safe summer, and thank you so much. It's been great being in class with you all. Aloha.